Chin. Okay, very good evening, my dear friends. Welcome to today's session. Today's our eminent resource person, Dr.A.KAVITA Madam Garu, Assistant Professor, Department of Botany, RBVRR Women's College, Autonomous, Hyderabad. Madam is going to explain about BRYOPHYTA. My dear students, listen carefully. Madam has a huge experience in this area. So CPGET students will utilize this wonderful opportunity. Madam, please start your session.
Yes, sir. Thank you very much. Hello, good evening to one and all. This is Kavita. I'm from the RBVRR Women's College. So today we are going to study with the first genera of your syllabus of CPGET and are related to bryophytes, that we call it as Marchantia. So already you might have studied this in your classes during your first year, but as per the point of your competitive exam, we'll just have a glance on the most important points of this genera. Not completely, but we'll have a glance like what it is, how it is, habitat and all that. Okay. So now when we talk about the systematic position, that is the classification of this Marchantia, it belongs to the class which we call it as Hepaticopsida. Order Marchantiales, Marchantiaceae, and Marchantia is a genera. So why it is called as a Hepaticopsida? So there's an important question in the exam point of view. So here when we talk about the thallus structure, so when you look into the thallus of this Marchantia here, the thallus is resembling just like the lobes of the liver. That is an animal liver. So the shape of the thallus will be resembling just like that. So hence it was included under the class Hepatica. Hepatica is always refers to the liver. So hence it belongs to the class Hepaticopsida. So remember this. And we can also call them as liver warts also. The commonly these are Marchantia genera. They are also called as liver warts. Another important point here. Next, so when we talk about some common genera which are found in India and the common species of Marchantia found in India, it includes Marchantia polymorpha, Marchantia palmata, Nepalensis, Marchantia indica, and Marchantia simlana. These are the important species which we are generally found in the Indian country. Now, so when we talk about where it is growing, it is a terrestrial bryophyte. Terrestrial, that means growing on the land surface, and that too on the moist shady place and the damp soil or rocks or the banks of the rivers. That means to say it is a general character of a bryophyte. All the bryophytes, they always prefer to grow in the moist conditions. Okay, and this Marchantia is a terrestrial one growing on the land surface. Now, and when we talk about the plant body, the thallus is referred to as the gametophyte, the dominant phase. This is another important point in the competitive exam. What is the dominant generation or the dominant phase of bryophytes? It is gametophyte. Okay. So here the thallus plant body is referred to as gametophyte. Because why it is gametophyte? Because the thallus is at the time of reproduction from the vegetative thallus, it is going to develop the reproductive organs, the so-called male reproductive organ, which is called as antheridophore and the antheridia, and the female reproductive organ called as archegoniophore and the archegonia. So as the reproductive organs, the male and the female, they are directly going to be formed from the adult thallus. Hence, the thallus plant body is referred to as gametophyte. Now, so when we go about the structure of the Marchantia, it is a dorsiventral thallus. So when we talk about this is the structure of the Marchantia, it is a dorsiventral thallus. Dorsal side is going to be differ from the ventral side based on the color and also some peculiar characters. Now, what are the characters you can see on the dorsal surface is on the dorsal surface, you can see the presence of gemmae-cups. This is the vegetative reproductive parts of the thallus plant body. They are helping in carrying out the vegetative reproduction. So in this gemmae-cups, we can see the vegetative bodies, what we call them as gemmae. They will reproduce. That is, they are like spore-like structures. When they detach from these cups, it have the capacity, the gemmae are having the capacity to develop into a new thallus. Okay, and you can observe here on the dorsal surface, the thallus on the dorsal surface, it is a dichotomously branched thallus. That means you can see the thallus is divided here into two. So we call it as dichotomously branched thallus. And on the central surface, on the central side, you can see a medial line or a central line, which we call it as a midrib. On this midrib, you can see the development of these gemmae-cups during favorable and unfavorable conditions. Now, at the time of reproduction, directly from this dorsal surface only, you can see the development of the male and the female reproductive organs. Now, when we come to the ventral surface of the Marchantia, so the ventral surface is dark brownish in color, and the dorsal side is green in color. It is dichotomously green. It is performing photosynthesis. Here we can see the pores also. On the dorsal side, you can see the presence of pores also, the small openings. So whereas when you go to the ventral side, it is dark brown in color, and we can see the presence of scales. This is another important point in the Marchantia. We can see the presence of the rhizoids and the scales, which are helping in fixing this plant to the thallus, to the substratum, that is to the land surface, or wherever the plant is present. These rhizoids and the scales, they will help in fixing the thallus to the surface. Now, when we go for in Marchantia, this is another one more important point here. We are going to find two different type of rhizoids here. One is called as a smooth-walled rhizoid. So here you can see these are the smooth-walled rhizoids, which are just like a tube-like. But whereas here, when you observe this, these are called as tuberculate. Okay, that means inside this rhizoid, you can see some outgrowths or these peg-like outgrowths, we call them as. Okay, so these are called as tuberculate rhizoids, and these are the smooth-walled rhizoids. So these are the two important points regarding the vegetative reproduction, and these rhizoids, they help in fixation and the absorption of water and the minerals from the substratum. Now, when I talk about the internal structure, so this plays a very important role here. So the internal structure of the thallus is differentiated into the three regions that we call it as the upper epidermal region and the middle photosynthetic region, storage region, and the lower epidermal region. Okay. So the epidermal region, when you talk about, it consists of the single layer of epidermis with the pores. So we already in the morphology, I told you, the thallus is comprising of small openings, what we call them as air pores. Okay, so that is observed in the epidermal region. And when we go for below this epidermal region, the cells are linearly arranged. You can see here, these are the cells which are linearly arranged one above the other, and these cells are forming the chambers. Okay, there are certain openings, that is, there is an empty space, or these all these cells are they are very loosely arranged, creating some empty spaces in between them. So we call them as the air chambers, and they are separated by this linear partition wall of the linear elongated cells. Now below this this is called as the photosynthetic region, or we call it as the assimilatory zone. or assimilatory filaments, because in these cells, in this region or in these cells, you can see the presence of chloroplast, useful for photosynthesis. So this is a region helpful for preparing the food material. So hence, as they consist of the chloroplast in them, the dorsal side of the thallus is green in color. Okay, and below this region is the storage region. We call this as the storage region on the ventral surface of the thallus. So this region is also consists of parenchymatous cells. They are very thin walled and they are very closely arranged with one another without any intercellular spaces. And this region, why I'm calling it as a storage zone, because these parenchymatous cells, they are helping in storing of the food material, the excess of food material or the protein or the granules, the starch granules, they will be stored in them. So hence it is referred to as the storage region. And below is the lowermost region, which we call it as lower epidermis. From this lower epidermis, you can see the emerging of the root hairs, or we call them as the rhizoids, as well as the scales. So this is regarding the internal structure or the anatomy of the Marchantia thallus. Now when I go for the reproduction, so generally we talk about the reproduction here of two types, that is a vegetative reproduction and the sexual reproduction. So the most important point here is the vegetative reproduction is carried through with the help of a specialized structures, what we call them as the gamecops. Now these gamecops possess the reproductive bodies, what we call them as gamete. These are the one which are stored. So generally when we talk about the structure of the gamete here, you can see they are a small globose structure. They are round in structure with a small notch at the center and having a small stalk. So these are the gamete. So generally a number of 8 to 10 gamete are present in a single gamecope. Okay, each gamecope may consist of around 8 to 10 gamete. Okay, so what is their role? These gamete are storing a lot of food material in it. Okay, so and they are serving as the spores. They are acting like a spores. So when these gamete, when they detach or when they come out of these gamecops, when they fall on a suitable substratum, they are germinating and producing the new thallus. So hence they play a role of vegetative reproduction. So there is a question in the competitive exam going on like this. What are the reproductive, vegetative reproductive bodies helping in producing a new thallus? So vegetative reproductive bodies. So here it refers to the gamete. Now next, when we go for the sexual reproduction, Marchantia reproduce sexually mainly by oogamous type. So what is oogamous? So there are three types of sexual reproductions: isogamous, anisogamous, and the oogamous type. So oogamous is such a type of sexual reproduction where the sexual organs or the reproductive organs, they both morphologically and physiologically, they differ from one another. That means the male sexual organ, structural wise as well as functionally, it is quite different from the female sexual organ. So hence we call it as oogamous type of reproduction. And Marchantia is a dioecious thallus. Okay, with reference to the reproduction, Marchantia is referred to as dioecious. What does this dioecious mean? That means to say the male reproductive organ is developing on one plant and the female reproductive organ is developing on the another plant. That is, male and the female thallus, they both are separately formed. They both are differing from one another. So hence the species or the Marchantia is referred to as the dioecious thallus. Right. So now when we talk about the male sexual organ, which we call it as a antheridia. Okay. Now, so these antheridia, now I'm talking about the structure of the male thallus referring to the male sexual organ. We call it as an antheridia. Now, so Marchantia is such a thallus which is recognized by the antheridia, or the sexual organs, both antheridia as well as the archegonia. They are developed on a receptacle, a stalk-like structure, which are called as receptacle. So this is another important characteristic feature of the Marchantia. Male and female reproductive organs developing on the receptacle. We call them as stalked sexual organs. In no other bryophytes you come across this point. Okay, so remember this. Marchantia consists of stalked reproductive organs, and these stalks or the receptacles, what we are talking about, if the receptacle is carrying the antheridia, then it is referred to as antheridiophore. And if the receptacle is carrying the archegonia, then we are calling it as archegoniophore. Okay, so these are the important points here. Now, so these antheridia and the archegoniophore, they are going to develop from the dorsal surface of the Marchantia thallus, and that too from the mid region, and they are projecting out of the thallus. Okay, they are not embedded, they are not present inside the thallus, they are projecting out of the thallus. So this is another important point. So whereas in the another genera, what we talk about Anthoceros, in that you can see the vegetative, the reproductive organs are embedded inside the thallus, but here it is not such case. So that points you need to remember. Next, now when we talk about the structure of the antheridiophore here, so the antheridiophore, that is the stalk of the antheridia, we consist of a disc-like structure. So the tip of this antheridiophore consists of a disc-like structure with air chambers alternating with the antheridial chamber. That is the topmost position of the disc is lobed. So there are around 8 lobes you can see at the position of this disc. Okay, so these discs, when you talk about the lobes here, one lobe, that is, it is going to, one lobe is of air chamber, and it is alternating. This is the air chamber alternating with the antheridial chamber. Now, what is this air chamber? So this is an empty space of the disc which consists of the assimilatory filaments or the photosynthetic filaments helping in preparing the food material and supplying the food material to the developing antheridia in this antheridial chamber. Now, what is this antheridial chamber? So this is a lobe which is going to possess the male sexual organ, which you call it as antheridia. So hence it is called as the antheridial chamber. So there are 8 such lobes or the chambers present in the disc of the antheridia. That is the disc of this antheridiophore. Now, so when we talk about the structure of this antheridia, they are globose structure. So this is a globose structure with a small stalk. And another important point of pertaining to the sexual organs in bryophytes are these sexual organs are covered with jacket layer of cells. All the sexual organs of all the genera, they are protected. They are covered with single or 2 layer of jacket layers. So that's a very important point. Now, so here this is an antheridial chamber in which you can see here this is a single antheridia globose structure having a single layer of jacket surrounding it, and this antheridial chamber is opened. It is not continuous here. we can see a small opening here of this antheridial chamber. We call this as the ostiole. Now, when you talk about the antheria, internal to the antheria, you can see the presence of the androcyte mother cells. So all these are the cells, what we call them as androcyte mother cells. Now, at the time of maturity, these androcyte mother cells, they undergo reduction revision and produce uninucleate, small, haploid, biflagellate gametes, what we call them as antherozoids. So I repeat, these androcyte mother cells on reduction division produce biflagellated. Okay, all the male gametes in bryophytes are motile. They possess two flagella, which we call them as biflagellated. And these, and we call them as the antherozoids. Okay, now when there's complete, this antheridia is filled with antherozoids, then the apex of the antheridia or the tip region of the antheridia, it will simply open and liberates all the antherozoids from this antheridial chamber, from this ostiole. So this is how the antheridiophore and the antheridia are going to, that is antherozoids are going to be formed. Now when we talk about the archegoniophore, which is called as the female receptacle, on which this is also here, when you observe the archegoniophore, it is appearing just like a flower, okay, having 8 lobes here also. It consists of the 8 lobes. And important point of Marchantia here to be remembered is as in video 4, the antheridia are developed inside the antheridial chamber. But here, the archegonia, they are developed in an inverted manner. So you can see here, these are the female sexual organs, which are called as archegonia. They are inverted in position, that too at the base of the disc. Okay, that is at the lowermost position or towards the inner side of the disc. You can see the development of or the production of these archegonia, and they are in an inverted position, hanging downward. Okay, this is another important point to be remembered in Marchantia. Archegonia are inverted. Okay, now when we talk about the structure of the archegonia here, so this is an archegonia, which is a flask-shaped structure having this globose venter. We call this as, this is a broad region, a globose region. We call this as the venter region, and long elongated part from this venter, we call it as the neck region. And this venter is of important here, consisting of the female gamete, we call it as egg cell, and comprising of it as a venter canal cell. And the neck region is going to consist of a row of neck canal cells. And on the top, so generally there are around 6 to 8 neck canal cells in the neck region, and the tip of this neck is going to be covered by 2 peculiar cells, what we call them as cover cells or the lid cells. So this is regarding the structure of the archegonium. So when we go for the fertilization here, from the male thallus, and that too from the antheridiophore or from the antheridia, the antherozoids, the male gametes, they are released into the environment, and generally at the time of rainy season, the antheridias will burst, and all the antherozoids, they are released into the rainwater. Okay, so These anthrozoids, along with the rainwater, they will be reaching the female receptacle, what we call it as the archegoniophore or the archegonia. Okay, where the archegonia is present. So along with the water, the male gametes are carried towards, you can look at the picture here. This is a water drop. So along with the water drops, the anthrozoids are carried. So you can see here, as they are biflagellate, they can swim in the water here. In this rainwater, these anthrozoids can swim. So that's why they possess flagella. So through this rainwater, they are reaching the archegonia. That is the tip of the archegonia, what we call it as the neck region. So from the neck, by the dissolving, so these anthrozoids, one more important point here, the neck canal cells and the lid cells, at the time of fertilization, they will get degenerate or they will dissolve. The neck canal cells and the venter canal cells, they will degenerate. As they degenerate, they will be oozing out some fluid, which is a source of attraction for these anthrozoids to reach the venter in which the female gamete is present. Okay. So as the water drop along with the anthrozoids, it reached the female receptacle. The archegonia are attracting the anthrozoids towards it, and these anthrozoids, they swim down towards the venter region, passing through the neck, and these anthrozoid, one of the anthrozoid will go and fuse. So this is anthrozoid, you can see here. It goes and fuse with the female gamete, the so-called egg. So the resultant of this fusion will be the zygote. Okay. So by the formation of the zygote, the gametophytic generation will end. Now, so next, the zygote is now developing into the next phase of the life cycle, what we call it as sporophyte. Okay. Now, what is the sporophyte here? So this is the structure which is developed in the archegonia or in the archegoniophore. So which is called as a sporophyte. Now, this sporophyte is generally differentiated into the three regions, what we call it as foot, seta, and the capsule. The zygote is developing into this structure inside the archegonia or in the female thallus. Now, the foot is the lowermost part of the sporophyte, which is in continuity with the female gametophytic thallus. Okay, and this foot, what I'm talking about, it is playing a role of getting the food material or absorbing the food material from the gametophytic tissue. So by this we can say that sporophyte in bryophytes here is dependent. Sporophyte is depending on the gametophyte for supplying of the food material, for getting the food material. So foot is the region which is absorbing the food from the gametophytic tissue. And next part coming up in the sporophyte is the seta region, which is just acting like a conducting tissue. So from the foot, the seta region will conduct the food material and the required water to the most important part of the sporophyte, the so-called capsule. So here in Marchantia, the capsule is a round globose structure and it is going to be covered by a single wall layer, and inside the capsule we can see the archisporium tissue. Now, or we call them as the archispore mother cells. Now, so these cells will undergo reduction division again, and resulting in formation of haploid spores. So these are the spores. Okay, and some of the cells will develop into spores, and some of these cells are developing into the elaters, the long elongated elaters. So these elaters, they help in dispersal of these spores from the capsule region. Now these spores, when the capsule wall breaks, all the spores along with the elaters, they are dispersed into the air. Now these spores, when they fall on the suitable substratum, they will undergo germination and develops into a new gametophytic thallus. So this is how a new thallus is going to be developed. This is a spore which is considered to be as the first cell of the gametophytic generation. And what is the structure here? It is the spore is going to be covered by a thick wall, what we call it as a spore wall. Now this spore, first it will undergo 2 unequal divisions, and later on one of these cells will keep on dividing and forms the gametophytic tissue, and the lowermost cell, which is undivided, it will develops into the rhizoids or the scales of the gametophytic tissue. So this is how the gametophytic tissue is formed. Now, when we look into the life cycle here, so this is a gametophyte, this is a male gametophyte, this is a female gametophyte here, and this is a male gametophyte. So female gametophyte here produces, that is in their archegoniophore, we can see the archegonia, and this is a disc which consists of the antheridia. So archegonia comprises of the female gamete, we call it as egg, and from the antheridia, the male gametes biflagellated. You can see here, these are the 2 flagella, and this is an anthrozoid. The male gamete, what we call as the anthrozoid. So these anthrozoids, they will swim in water, pass through this long neck, and reach the egg. They reach the venter, fuses with the egg, fertilize, and forms a zygote. So this zygote will undergo several repeated divisions, and forms the sporophytic tissue, what we call it as the sporophyte, which is the a globose long structure which is dependent on the gametophyte. So from the sporophyte here, spores are going to be released by the reduction division. So these spores, they will form. So there are many spores. So by reduction division, for each sporophyte mother cell will form, each sporophyte mother cell will form 4 spores. Out of this, 2 spores may form, or 3 spores may form male gametophyte, and 1 may form female, or 2 spores may form male and another 2 spores may form female. It depends upon the sexuality. The spores are developing further into male gametophyte and the female gametophyte. Okay, I hope this is clear for you all. So thank you. I'm just stop sharing. So if you have any doubts, please, you can. Any doubts regarding this in the thallus structure, the gametophyte and the sporophyte of Marchantia? I hope this is clear, sir.
Any doubts? So madam has clearly explained about the entire morphology and anatomy, reproduction, life cycle of Marchantia. Any doubt? Is there any doubt, my dear friends? Please ask our respected.
I have also highlighted the important points in the exam point of view. Right. Okay, sir. Thank you.
Okay, madam. Thank you. Tomorrow?
Yes, sir. Tomorrow I'll start with anthocirrhosis.
Okay, madam. Thank you.
Thank you.